The goal of this experiment is to replace a canonical amino acid by a non-canonical amino acid for recombinant protein production by selective pressure incorporation to produce the novel gold fluorescent protein.
The method of selective pressure incorporation is intended to produce proteins with novel chemical features. This is especially useful when attempting to understand or to engineer protein function.
The main advantage of this technique is that it is relatively simple to conduct, because it is similar to common recombinant protein production.
Time and wavelength correlated fluorescence is helpful to investigate the excited state dynamics of the modified protein, which in turn makes it easier to understand the influence of the non-canonical amino acid introduced into the chromophore on the optical properties.
SPI can, in principle, be applied to any protein production setting for which auxotrophic strains and chemically defined media are available. This includes other bacterial strains and also mammalian cell culture.
The timing and the setup of expression conditions are critical, since it's mandatory to suppress the production of the non-modified wild-type protein. That's why certain parameters such as the temperature, the time of induction, or the composition of the cultivation medium need to be optimized.
We first had the idea for this method after initial studies of protein X-ray crystallography with metal-containing amino acids such as selenium-methionine. Demonstrating the procedure will be Almut Pelzer, a student from my laboratory.
To begin this procedure, transform tryptophan auxotrophic E. coli cells with the expression plasmid for eCFP.